So today we're diving into something that is, it's really the bedrock of modern genomics.
Mm.
These massive, sprawling, single-cell reference atlases.
Absolutely. Things like the Human Cell Atlas. We're talking about these colossal databases with millions, sometimes tens of millions of cells covering every tissue you can imagine.
They're supposed to be the ultimate map of human biology, right? The Google Maps for our cells.
That's the dream, a common coordinate system where any lab, anywhere in the world, can take their new data, map it onto this reference, and immediately understand what they're looking at.
But, of course, it's never that simple. The reality is that for most individual labs, these incredible maps are basically unusable. They're locked away in these data silos.
Right, and there are a couple of huge reasons why. The first is just the raw computational power required.
Yeah, you can't just download the whole Human Cell Atlas and run it on your laptop. You need the expertise and the hardware to rerun this gigantic, complicated integration pipeline with millions of cells just to see where your, say, 5,000 cells fit in.
And that's before you even get to the stickiest problem in all of single-cell biology, the batch effect.
The infamous batch effect.
It's the technical noise. Every lab uses slightly different machines, different chemicals, different protocols. And that variation, that technical noise, can completely drown out the real biological signal you're trying to find.
And traditional methods for removing that noise? They can be a bit clumsy, can't they?
They can be disastrous, actually. If you have a big biological change, like a disease that affects almost all of your cells, the algorithm can get confused. It might look at that huge biological signal and mistake it for a technical batch effect.
And it just erases it.
It erases the very discovery you were hoping to make.
So that's our mission for this deep dive. We need a way to break open these silos. We need something fast, scalable, decentralized that can handle batch effects without destroying the biology.
And that's exactly where the solution we're focusing on comes in. It's called single-cell architectural surgery, or for short, scArches.
Architectural surgery. I like the name. It sounds precise.
It is. The whole point of scArches is to allow a lab to quickly and accurately map their new small data set onto a huge reference atlas without ever needing the raw data from that atlas. It's designed for a world where data can't always be shared.
Okay, this is the core of it. And the central idea here, it's not actually new to biology, is it? It's a concept borrowed straight from the world of AI.
That's right. It's all about transfer learning.
Transfer learning. This is what blew the doors open for things like computer vision and language models.
Exactly. Think about models like BERT for language or ImageNet for pictures. You don't train a new model from scratch every single time. That would take weeks and a mountain of data.
Right. You take a model that's already been trained on, millions of images and already knows what edges and shapes and textures look like.
And you just reuse that knowledge. You transfer it. You fine-tune that massive pre-trained model for your specific smaller task. scArches is basically the first really successful application of that same idea to genomics.
So how does that work mechanically? How do you perform surgery on a neural network?
Okay, so you start with a base model. scArches uses a type of deep neural network called a conditional variational autoencoder, or CVAE. You might have heard of models like SCVI or TFAE. Those are CVAEs.
Okay, hold on. CVA. Can we break that down? That sounds like a technical minefield.
Let's use an analogy. Imagine your single-cell data is like a photograph of a cell, but every lab is using a different camera. Some are old film cameras, some are fancy digital ones. The lighting is different in every lab. That's your batch effect.
All that technical noise.
Exactly. The CVAE's job is to act like a master photo editor. It looks at all these different photos and learns to see past the camera type, the lighting, the focus, all that noise. It learns to extract the true underlying image of the cell itself.
So it's learning to separate the technical junk from the real biology.
Precisely. It takes all that high-dimensional gene data and compresses it down into a much simpler, low-dimensional map. We call this the latent space. And because it's conditional on the batch label, the camera it came from, it learns to build that latent space so that the back effects are gone, cells of the same type cluster together no matter which lab they came from.
And that latent space, that clean map, becomes our reference atlas.
That's our reference.
Okay.
So now we have our beautiful pre-trained reference model, and you come along with your new small data set from your lab. This is where the surgery happens.
Okay.
The first thing we do is we take that huge reference model and we freeze almost all of its weights. We lock them down. All that knowledge it learned about basic cell biology and common batch effects, it's preserved. untouchable.
Because you're not retraining the whole thing.
Not even close. The surgery is incredibly minimal. All scArches does is add a few new input nodes for your new study's batch label, and it attaches a tiny, tiny set of new trainable weights. They're called adapters.
Adapters. It sounds like you're building a small, custom-made porch onto a massive pre-existing mansion.
That's a perfect analogy. The mansion, the core biological knowledge, remains untouched. You're just training this tiny little adapter to understand the specific technical quirks, the camera settings of your new data set.
So the number of parameters you're actually training must be minuscule in comparison.
It's a staggering difference. You're reducing the number of trainable parameters by 4 to 5 orders of magnitude. That's a factor of 10,000 to 100,000.
Wow. Okay, so that's where the efficiency comes from. That's a huge shortcut.
And speed up is incredible. It's about a 5 to 8-fold speed increase over doing a full integration from scratch. You can map 1 million new cells in less than an hour on a standard machine.
Less than an hour. That takes it from a specialized high-performance computing task to something any lab can do routinely.
That is the entire point.
So quick recap then. We have these giant siloed cell atlases. The solution, scArches, uses this idea of transfer learning. It takes a pre-trained reference model, freezes most of it, and then trains just a tiny set of adapters to quickly map in new data.
And the efficiency gain is just massive.
Okay, so let's ask the question you're probably thinking right now. If we're only training this tiny fraction of the model, does the quality suffer? Are we sacrificing accuracy for speed?
That is the critical question. And to answer it, you have to measure 2 things at once. First, did we actually remove the batch effect? And second, did we preserve the real biology?
Right, you need to succeed on both fronts.
For batch removal, we use metrics that check if the same cell types from different batches are mixing well together. And for biological conservation, we check if the distinct cell clusters we knew about are still perfectly separated after we integrate.
And the results?
The results show that this minimal fine-tuning strategy performs competitively with the full, from-scratch integration methods. The accuracy is there.
Okay, let's talk about some of the hard cases, the places where you'd expect a shortcut method to fail. What about really rare cell types?
An excellent test, and scArches performed really well. In one pancreas dataset, for instance, it successfully mapped and preserved a tiny population of delta cells.
And how rare are we talking?
They made up about 0.5% of the entire dataset, so really a needle in a haystack, and the model found it and placed it correctly.
What about something more dynamic, like cells changing over time, a continuous process like differentiation?
It handled that beautifully, too. It was tested on data from pancreatic development, where you have cells differentiating into their final fates. scArches preserved those smooth, continuous trajectories perfectly. It didn't just lump them into disconnected blobs.
And the ultimate test, telling apart 2 cell types that are almost identical, the classic example being natural killer cells and NKT cells.
Right. For immunology, that's a huge deal. They ran a test where the reference atlas had NKT cells, but it had never seen NK cells before.
So the query data contained a totally new but very similar cell type.
Exactly. And scArches placed the new NK cells in their own distinct cluster, but crucially, it placed that cluster right next door to the NKT cells in the map. It understood their biological similarity without merging them. It shows a really nuanced understanding.
That's incredible. So it's not just integrating data, it's actually transferring knowledge. Which brings us to the next step. What kind of knowledge can we transfer?
Well, there are 2 main types. The first is discrete labels, which is basically just automated cell type annotation.
Tell me more.
So there was this great example where they mapped a query data set of about 90,000 mouse cells onto a massive reference atlas called Tabula Senis. The goal was simple. Could the model automatically assign the correct cell type label to the query cells?
And how did it do?
It achieved about 84% accuracy across 24 different tissues.
84% is pretty good, but it's not perfect. What happens with the other 16%? Does the model just take a wild guess and potentially get it wrong?
And this is maybe the most important feature for anyone listening to this. The model doesn't just give you a label, it gives you an uncertainty score.
Yeah.
If the model is not confident, if the uncertainty is over 50%, it labels the cell as unknown. It knows what it doesn't know.
So it's not forced to make a bad call.
Better than that. In that experiment, they intentionally included query cells from a tissue the reference had never, ever seen before, the trachea. And the model correctly labeled almost all of them as unknown. It recognized they didn't belong anywhere on the existing map.
That is huge.
Yeah.
That prevents so many downstream errors, so it knows its own limits.
That uncertainty score is arguably as valuable as the accuracy itself.
Okay, so that's discrete labels. What about transferring more complex continuous information? You mentioned modality imputation.
Right, so this is where you combine scArches with another model called Total 6. Total 6 is designed for multimodal data specifically. Data where you have both RNA expression and surface protein levels from the same cell, which you get from a technique called CIT6.
Which is really powerful, but also expensive and relatively new. Most of the data out there is RNA only.
Exactly. So the workflow is this. You build your reference atlas using rich CIT-seq data that has both RNA and protein. Then you take an older, cheaper RNA-only dataset as your query.
And you map the RNA-only data onto the multimodal reference.
Yes. And because that reference map, that latent space, understands the relationship between RNA and protein, the model can actually impute the missing protein levels for your query cells.
So you can predict what the protein expression would have been on cells where you only measured RNA.
You can. You can predict levels of key proteins like CD3 or CD14, effectively adding a whole new layer of data to older experiments, which is just transformative.
So let's recap again. We've seen scArches is fast, it's accurate even with rare or similar cells, and it can transfer knowledge, both simple labels with a crucial uncertainty score and complex continuous data like protein levels.
Which brings us to the highest stakes application of all.
Using it to understand disease.
Exactly. Contextualizing disease data against a healthy reference is the ultimate goal. But it's tricky. You need the model to do three things perfectly.
Okay, what are they?
1, it has to preserve all the normal variation in the healthy cells. 2, it has to correctly integrate the cells from the disease sample that match healthy cell types. And 3, and this is the most important, it has to preserve the distinct variation caused by the disease. It can't erase it.
And the big case study for this was COVID-19. They mapped lung samples from COVID patients onto a healthy immune atlas.
They did. And the key was in how they set up the training. The reference atlas was built only from healthy tissues, bone marrow, blood, normal lung. The model never saw the COVID-19 biological signal during its initial training.
So when the COVID data was introduced as a query, that disease signal was treated as totally new, unseen biology.
Exactly. The frozen part of the model handled the basic cell identities, the little adapters handled the new technical batch effect, and the unique disease signature was just left alone, allowed to stand out.
And what did it find? What did the final map look like?
It was a huge success. First, the matching cell types, like normal tissue-resident macrophages, they integrated perfectly with their healthy counterparts. But, crucially, the model preserved the emergent disease states.
What does that mean, emergent states?
These were cell populations that simply didn't exist in the healthy reference. For example, it identified a population of inflammatory macrophages that came from monocytes, we call them MOAMs, that were found almost exclusively in patients with severe COVID-19.
And it didn't try to merge them with the healthy macrophages.
Nope. It placed them as a distinct group close to the monocytes in the reference, correctly reflecting their developmental origin. It discovered a disease-specific cell state.
That's incredible. Did it also pick up on more subtle changes?
It did. Even the normal macrophages from patients with moderate COVID didn't totally mix in with the healthy ones. They formed their own little neighborhood on the map because the model preserved their activated state, marked by inflammatory genes like IFI27 and CXCL10.
So it's sensitive enough to preserve not just new cell types, but also subtle activation states within existing types. It's a true biological filter.
It is, but we have to be clear, it's not a magic bullet. It has limitations.
Of course. What are the big ones?
Well, first, it inherits the limitations of its base models. For example, the output is that low-dimensional latent space, that map. It's not a corrected gene expression table. You can't just export a clean spreadsheet to use with older tools.
And for a lab that wants to start using this tomorrow, what's the biggest practical constraint?
The quality of your reference. The whole principle of transfer learning relies on having a good, comprehensive, diverse reference to start with.
So you can't just use a reference built from one single experiment.
Please don't. That's the biggest mistake you can make. The reference needs to be built from multiple studies, multiple batches, so the model learns to be general. Also, you need good overlap in the genes you're measuring between your query and the reference. It's robust to some missing genes, but it works best when the feature sets are highly similar.
Even with those caveats, this really feels like it changes the game for collaboration.
It fundamentally changes it. International groups working on, say, a cancer atlas, they face huge legal hurdles with sharing raw patient data because of rules like GDPR or HIPAA.
But with starches, you don't need to share the raw data.
You just share the model weights, these compact little files that contain all the learned knowledge, but none of the sensitive patient data. It allows for decentralized iterative atlas building in a way that just wasn't possible before.
So if this is the bridge from big data to actual results, what's the future vision? Where does this go next?
The immediate future is building large-scale disease atlases. Imagine being able to take data from a mouse model of a tumor and instantly map it onto a reference of thousands of human patient tumors to see which mouse model is actually the best mimic.
That would accelerate drug development enormously.
And longer term, the vision is for fully multimodal atlases, integrating not just RNA and protein, but also epigenetics, chromosome structure, spatial data, all into one unified, queryable map of human biology.
It's what makes scArches so important. It turns these atlases from static academic artifacts into living, dynamic tools that anyone can contribute to and use.
Exactly. By letting us share the model instead of the data, it completely rewires how we can do collaborative science, especially in clinical settings.
And that brings us to our final provocative question for you, the listener. If these research models become shareable assets, easily updated and refined by anyone in the scientific community, what critical scientific collaboration that's impossible today could become routine next year? What field, be it neurobiology, cancer, infectious disease, what field would you choose to build the first truly shared, community-owned reference atlas? We'll leave you with that thought.